next one up, I'm very excited to ask to the stage Sam, who's already over here. So Sam is not only hosting us graciously tonight for the speaker and sponsor after show party, but you are also, let me just download your slides in case there's any images that should be showing correctly. Sam is also working at Future House. Who here has heard of Future House already? Okay, well, you're really preaching to the choir. That is quite a lot of people. They're not very old, but in a very, very short amount of time, they've really managed to produce a few fantastic output. And I told you earlier there that one of the things that we're very excited about at Foresight is AI for automating scientific discovery and for scientific research, and that is very much what Future House is up to these days. So thank you so much for joining us, Sam. I also first met you in person at Burning Man by stumbling out of my tent, and you were my tent neighbor. So that is another fun fact of that. And you will understand how, because Future House tonight very much is inspired by the principles of Burning Man. All right, well, without further ado, thank you so much for joining us, Sam. Thank you so much for hosting us tonight. It's a real joy. Please take it away.
Great, thank you so much. Which one? Oh, great, okay, yeah, sorry, I'm very loud without the microphone, so. Okay, so this is another talk where we have a lot of stuff to get through and not very much time, so we're just going to dive in. So I run Future House. We're a new AI for science research lab, and we're focused on automating scientific research. And so basically, the thing you need to know is that biology is limited by intelligence, right? There are tens of thousands of different cell types in the brain. There has been individual research done on each cell type. You don't have enough time. No neuroscientist has enough time in their life to go and figure out, like, read all the literature and understand what role each cell plays and how the brain works, right? Similarly, like, if you want to go and understand how the cell works, there are 20,000 genes in the human genome. You don't have enough time to go and read all of them and understand what role each of them plays, right? And so this plays out in this example. This is an example of how this plays out and the significance of this. Matthew Kirshner does drug discovery, and he had this tweet, which I really liked, talking about how he started Trace Neuro, which is a company that raised $100 million, and how they did it, right? And basically, the trajectory that led to founding this company, it's pursuing a therapeutic for ALS, is to start with they start with this protein, TDP43, which is well known for its role in ALS, and it regulates splicing. And so then they realized, we should look at what the downstream consequences are of the splicing defects. And so they went and they basically looked at all 66 different proteins that are regulated, where the splicing is regulated by TDP43. They found that some of them have human genetic data that links it to ALS also. They identified the specific variant in that protein that is linked with the disease. This is all in silico exploratory research. It probably took them 6 months to follow all these breadcrumbs, maybe a year or something like that. And my thesis is that there are lots and lots of examples of this where there could be drugs. There are drugs that we could be making. There are insights that we could have if only someone had thought about it, right? And so this is what I mean when I say biology is limited by intelligence. And the question that you should then ask is, what happens if we have an infinite number of expert biology researchers who can then just go and do this at scale, right? And so this is our mission. It's to automate scientific research. Here are some key facts about us. We're an independent nonprofit. We're based in San Francisco. We're over in the Dog Patch. And for anyone who's coming tonight, we're super excited to host you guys. We do have a cool space. It's going to be great. We launched about a year ago. We're about 15 people total. We're primarily funded by Eric Schmidt, although we are very lucky to have a bunch of grants and contracts from other people as well, including Open Philanthropy, for example, and some other organizations also. And I also like to put up here, this is really important. People ask me why we're nonprofit. I'm up here talking about target discovery and et cetera. The rate at which things are progressing, it is, I think, very actually feasible that maybe in 2025 might be a stretch, but in 2026 for sure, the vast majority, the majority of all good scientific hypotheses that are generated, may be generated by us or by systems that we're building or by the systems that look like the systems that we're building, right? Scientific discovery, I think, is actually very soon going to become largely automated, and when that happens, I think it is very important that the vast majority of that knowledge makes it out into the open, which is why we're nonprofit. Obviously, also there are some insights that you can only have impact if they're proprietary. Like if you discover a drug, you have to patent it. If you can't patent it, you can't invest in it, and then it can never make its way to market. And so we do think about spin-outs and stuff also. But I'm just going to move kind of quickly because we have data, and no talk is complete without data, and so I want to get there. But this is basically what the schematic for the thing that we're building. We're building an AI scientist, and what that means is we build a system where it can take some high-level quest, right? Based on that quest, like, for example, find a new drug target for ALS. Based on that quest, it can go and gather data from wherever. It can gather data from clinical trials, from databases, from the literature. It can analyze it. Based on its analysis, it can go and generate hypotheses, and then ultimately, those hypotheses it can propose for lab validation. We do have a wet lab, and lead to some discovery, right? I mean, the lab part of this, I will just say, the lab part is really important. There's no such thing as a discovery in biology without lab validation. So, okay, so this is what we're trying to build. So let's just move on. Here we go. Okay. So the first thing that you should ask when I tell you that we're going to automate science is you should say, well, science is like very nebulous, right? How do you know if you're succeeding? And so the first thing we did was to build a benchmark for the, I bet I can do this. Hold on. There we go. The first thing that we did was to build a benchmark to measure the performance of AI, AI agents, these AI scientists on scientific tasks. And it looks like this. It's called LabBench. It's published. You can go and look at it. We're also pretty proud of the fact that the frontier labs now use it for their evaluations. But basically, this is a benchmark that measures the performance of AI systems on a variety of kind of like standard tasks that you have to do when you're doing research in biology. And just to give you an example of one of them, this is a single example of what we call FigQA. It's a question answering benchmark for understanding scientific figures. And the question here is, what is the width of this entire image? Right? Now, the width of the entire image is not written there, but you can, but there are these various distance measurements that the people making this figure have put in for these holes that they've drilled. And based on those distance measurements, you can infer what the width is of the entire figure. This is just one kind of thing. It's like, when you're looking at a scientific paper, there are figures, and you have to be able to make complex, non-trivial inferences from those. So we have a couple hundred questions like this as part of the benchmark. There are 2,000 questions total in the benchmark. And so then you can ask, okay, well, how well do the models do? And I'm going to apologize. This slide is like woefully out of date, which means that I think it's from July. We're measuring the precision of the models here on all of the lab bench tasks. The green horizontal line at the top is humans. And then there are all the various models. The upshot is the models are really not very good at any of these tasks. And there are a few important caveats that this actually dramatically overestimates the performance of the models on these tasks in two ways. The first way is that this is precision, which means that the model has an opportunity to say it doesn't know. And if it doesn't know, you don't count it in this benchmark. So actually, the models usually only get about 5% of lit QA questions right. It shows up here as 45% because they refuse to answer 90% of the questions, and so that doesn't get counted, right? And so we have an accuracy, a total accuracy figure as well, and it doesn't matter, it's not here. So that's the first thing. The second thing is that this is multiple choice, and often when they do get it right, you can tell based on the reasoning they get it right by guessing. And that if you ask the same question in open answer format, the accuracy goes down by a factor of 5 or something like that. In fact, OpenAI has started to do that because O1 was human level on the multiple choice version of Protocol QA, and they just for the model they released the other day, they went to the open version, and it's really bad. So okay, the other reason why this is not fair. Sorry. Yes. We'll be done in a minute, I promise. The other reason why this is not a fair evaluation is because you need tools to answer many of these questions. And when we're doing this evaluation here, the models don't have tools. So the rest of what we do at Future House is basically building AI systems that have access to tools and then measuring the performance of those AI systems on some of these scientific tasks, right? And I just want to show you what this looks like. So this is Paper QA, which is a system that we built that has access to tools for searching the scientific literature so that we can see how well AIs do at searching the scientific literature when they have access to tools. And here what it did is you ask it a question. Now it's going at searching for a bunch of different papers, right? It finds relevant papers. When it turns green, that means that it has parsed the paper. And now it's going at summarizing each individual paper to identify relevant chunks. And it comes out, and because this is an agent, agent means that it sees the result and then it gets to do something, it gets to make another decision. Basically, it will go through many rounds of this process of searching for papers, reading the papers, searching for papers, reading the papers. It'll do this autonomously, right, until it generates an answer. And at each stage, what it's doing is it's saying, oh, this is what I found in the previous, from the papers I just found. This is what the papers I just found said, let me change my search terms, search again, let me search for this other thing instead, etc. Okay, so how well does it do? When the model doesn't have access to tools and you evaluate it on LitQA, which is a benchmark of retrieving information from the literature, you get like 44%, which is way below humans. When you give it tools, Paper QA2 there on the left, 85%. So these models actually exceed human performance at scientific literature search when you give them access to tools. This is why I say that within a year or two, I think it is very likely that we will be, that the majority of good hypotheses generated by the scientific community could be generated by us or by systems that are like this. Okay, I have no time, and so I'm just gonna say, basically, what do you do with this? Well, there are 20,000 genes in the human genome. There were previously only Wikipedia articles for 3,000 of them, and so we were like, well, we can do literature search at infinite scale, so let's just generate the other 17,000. That takes 2 days. It's great, and then you measure, you evaluate those articles. You have PhD students and postdocs go in a blinded format, evaluate the accuracy of those articles, and you find that the articles that are generated by the AI have half as many errors in them on average as real, actual Wikipedia articles for the same genes that are written and curated by humans. Okay, so that's cool. It's online. It's called wikicrow.ai. You guys can look it up. The other thing, if you want to play with the system, you should go to hasanyone.com. We put out a version of Paper QA that is freely available for answering the question, has anyone done something before? Because when you're a scientist, it's a pain in the ass. You ask that question all the time. And so you can go and look it up, hasanyone.com. It's another fun way to play with this. This is all also open source, so you can download it for yourself and etc. The last thing I'll show you before Allison kills me is that's just like literature search. You can give the models tools to do all sorts of other things as well. This is not public. This is called Protein Crow, and basically what this is what happens if we give the models the tools that they need to go and design new proteins. And so you can ask it a high-level question like, build me a binder against PD-L1, which is a protein that's important in a bunch of cancers, and it will go and it can use inverse design tools. It can go and use binding models, and it's autonomously going through cycles of generating proteins, looking at how good the proteins are, with various simulators, adjusting its parameters, doing it again and again, and at the end, it actually produces some proteins that do actually work in the lab. And we entered it into a competition, and it got position 60 out of 730, which means that there were 670 human submissions that were worse than this. Thank you, I'm done. Thank you, good.
Rudolph is gonna kill you. Thank you, Sam. Unfortunately, you used all of your question time, but you'll be here today, and definitely for anyone joining tonight, you'll be probably swarmed at that point. Thank you so much, and thanks for showing new research. Amazing.